Heredity is the backbone of how living things change over time. It's the process by which parents pass on a basic body plan and characteristics to their offspring. And those inherited features provide the starting point for variation and evolution. When organisms reproduce, they pass along a basic design or blueprint to their young. But that design isn't fixed. Each generation carries small differences compared with the one before. So when offspring reproduce, they inherit the parental traits, plus any new changes that have arisen. And over many generations, those small changes accumulate. The amount of variation depends on how organisms reproduce. Asexual reproduction produces relatively few differences because variations mainly come from copying errors or mutations. Think of sugarcane or bacterial fission, where individuals look very similar. Sexual reproduction creates much larger variation because of processes like crossing over, chromosome segregation, and mutation, which is why animals and humans show a wide range of traits. Not every variation has the same chance of surviving. Environmental conditions favor some traits over others. For example, during a heat wave, bacteria with heat-resistant traits survive better than those without. In other words, the environment selects which variations are more likely to persist, and that selection drives evolutionary change. Heredity is simply the transfer of characteristics from parents to offspring through reproduction. It's the reason traits such as skin color, hair type, eye color, and height can appear repeatedly across generations, and why family resemblances are common. When we talk about inherited traits, we mean features passed down from parents, like eye color or skin tone. Genetic variation means offspring can inherit traits in different combinations. A child might show a mix of parental features. A simple example is earlobes. If both parents have free earlobes, their children are likely to have free earlobes as well, because that trait is commonly passed on. Gregor Mendel laid the foundations for understanding how traits are inherited. He studied pea plants, focusing on traits such as seed shape and plant height, and used controlled crossings where he varied only one characteristic at a time to see how it was passed to the next generation. In one set of experiments, Mendel crossed plants that differed in height. A homozygous tall plant with genotype TT and a homozygous short plant TT. All of the first generation or F1 offspring were tall with genotype TT, showing that tallness was dominant over shortness in that cross. When Mendel let the F1 plants self-pollinate, the resulting F2 generation showed a predictable pattern. Genotypically, the ratio was 1 to 2 to 1, 1 TT, 2 TT, and 1 TT, while phenotypically, the plants appeared in a 3 to 1 ratio of tall to short, because both TT and TT are tall. He also crossed plants for two traits at once, such as seed shape and seed color. A cross between a round yellow plant, RRYY, and a wrinkled green plant, RRYY, produced F1 offspring that were all RRYY, indicating round shape and yellow color were dominant in those combinations. When those dihybrid F1 plants were allowed to self-pollinate, the offspring followed a 9-3-3-1 phenotypic ratio. 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Those numbers reflect all the possible trait combinations that result from independent inheritance of the two genes. Mendel's conclusion from these experiments was that different traits are inherited independently of each other, the principle of independent assortment, and that predictable ratios like 9 to 3 to 3 to 1 in a dihybrid cross arise from the random combination of alleles during gamete formation. Expression of a trait begins at the gene level. A gene's DNA sequence encodes a protein or enzyme. If the gene produces an efficient enzyme, that can trigger higher production of a hormone or metabolic product leading to a particular trait like tall growth. If the gene is altered and produces an inefficient enzyme, the downstream product may be low and the trait, such as height, can be reduced. At the cellular level, inheritance is organized on chromosomes. Most body cells are diploid, meaning they have two copies of each gene, one from each parent, noted as 2n. Germ cells or gametes are haploid, n, and carry only one copy. Fertilization restores the diploid state in the zygote, maintaining species stability through generations. Sex determination can be influenced in different ways depending on the organism. In some species, the environment, like incubation temperature, determines sex. That's temperature-dependent sex determination. In others, sex is determined genetically by specific chromosomes or genes, rather than by environmental conditions. In humans, sex is determined by chromosomes. We have 23 pairs of chromosomes, and the 23rd pair are the sex chromosomes. The mother always contributes an X chromosome in her egg, while the father's sperm can contribute either an X or a Y. If the fertilizing sperm contributes X, the zygote will develop as female. If it contributes Y, the zygote will develop as male. That's it for today. Learn smarter with EduRev. Notes, tests, and practice in one app. Download EduRev now on Play Store and App Store and keep learning every day.